We are preparing for the SeekSpace single-cell spatial transcriptome seq experiment. Now, let's get started. The first step is to prepare our nuclei suspension from the tissue slice and get them labeled with spatial barcodes. After fixation, both spatial barcode areas are processed sequentially for washing and nuclei extraction preparation. First, remove the fixation solution from each spatial barcode area. Wash each spatial barcode area once with 1 times PBSRI to remove residual fixation solution. Add NES buffer sequentially to each spatial barcode area. The chip is now ready for nuclei extraction on the single-cell suspension system. Set up the single-cell suspension preparation system on a stable stand and ensure the device is properly leveled before use. Transfer the NES buffer on the tissue into a microcentrifuge tube. Then remove the space chamber to expose the tissue section. Align and attach the silicone chamber, gently pressing to ensure a secure seal. Add fresh NES buffer to fully cover the tissue and prevent drying during processing. Place the chip on the stand and adjust the probe tip to sit 1 to 2 millimeters below the liquid surface without touching the tissue. Start the program and gently move the chip during operation, focusing on tissue-rich areas. After processing, collect the nuclei suspension and residual tissue debris into a collection tube containing NES buffer. Rinse the chip with NES buffer. If residual tissue remains, repeat the extraction steps as needed. Insert the ultrasonic probe into the sample tube and keep the tip fully immersed in the suspension. Gently move the probe within the liquid for 9 to 15 seconds to further dissociate remaining tissue fragments. After processing, return the tube to ice immediately. If visible tissue fragments remain, transfer the suspension to a fresh tube, add nuclei extraction buffer, and repeat the handheld sonication step. Combine the nuclei suspensions collected from repeated dissociation steps into a single 5 milliliter tube and keep the sample on ice. Repeat the same workflow for the second spatial barcode area and combine the recovered nuclei suspension into the second collection tube. Pre-wet a 40-micron cell strainer with wash buffer before filtration. Add wash buffer to the collected nuclei suspension and gently mix by pipetting. Pass the suspension through the strainer to remove large debris. Rinse the original sample tube with additional wash buffer and recover the remaining nuclei through the same strainer. Collect all filtrate into the same tube for downstream processing. Centrifuge the filtered nuclei suspension under cold conditions to pellet the nuclei. Carefully remove the supernatant without disturbing the pellet. Resuspend the nuclei in 1 times PBSRI and adjust the final volume to 190 microliters. This ensures that at least 175 microliters of nuclei suspension remain available after quality control for downstream processing. Keep the nuclei suspension on ice and repeat the same procedure for all remaining samples. For pre-RT, the total number of nuclei in a labeling region should not exceed 240,000. The nucleation rate should be above 5%, as lower rates can reduce the number of captured cells and genes. Aggregation should be less than 30%, since excessive clumping affects droplet encapsulation and single nucleus resolution. Acceptable nuclei diameters range from 5 to 40 micrometers. Spatial labeling of nuclei is complete. Next, we will proceed to the preliminary reverse transcription reaction.